Thank you very much for the introduction. And as mentioned by Ken, so this was recently published in JAV, and we were also lucky to have Ken as the editor. And I'm also really happy to have been invited to talk about this today and tell you about the exciting stuff we're doing with body fluids, and today specifically with saliva. So this is largely about Zika virus. As you can imagine, SARS-CoV-2 is more of a last-minute addition to this piece. So simply because Zika has been out of the news for a while, I'll give you a very brief, basically a reminder about Zika virus again. So Zika virus is actually not a completely new virus, and it wasn't either during the re-emergence in 2015, but it's a re-emerging virus, and it has already been discovered in the 1940s in Uganda, and it has since then been endemic to certain regions of Africa and Asia, and is also still present in these countries, but mostly gained notoriety during the re-emergence in Brazil in 2015. And it is a flavivirus, and as such pretty small and enveloped in a positive sense RNA genome. And as a flavivirus, it is also an arbovirus, and this means that, like its closely related, basically brother dengue, and also the more distantly related yellow fever virus and West Nile virus, it is largely transmitted by mosquitoes. And in the case of this is mostly Aedes, and most notably Aedes aegypti, but also Aedes albopictus, as you can see down on the left. And there are also very limited cases of non-arboviral transmission suspected to be sexually transmitted, and we will get to that specifically in a minute. Okay, so pathology-wise, it's pretty standard for flavivirus, so it causes the typical fever and rash that is also associated with dengue virus and other flaviviruses. Unlike those flaviviruses, there are also severe complications that are basically encountered in neonates or in utero in pregnant women. And this concerns severe developmental defects such as microcephaly, but also more severely fetal demise. And adults are generally okay and have only an asymptomatic disease, but there are also incidences of an autoimmune mediated muscle weakness called Guillain-Barré syndrome. Now, already having mentioned the African strain discovery in the 1940s. So this was 1947 when the African strain was discovered. And shortly thereafter, it was also noticed that it can be transmitted to humans. And then it took a few years until it also realized that there is an Asian lineage strain. And this was first isolated from a mosquito in 1966. And this is actually also the strain that has then also been further transmitted basically east, eventually to cause minor outbreaks, first in 2007 in Micronesia and on the island of Yap, later in French Polynesia, and then eventually the larger outbreaks in Brazil and generally the Americas from 2015 onwards, so of course, causing tens of thousands to hundreds of thousands of infections. Actually, it's probably a pretty good question, and most of you might be asking yourself what's going on with Zika, and the press has been slow about this, so there's not as much news about Zika virus, not only because of overshadowing by SARS, but generally. So there's still low levels of ongoing transmissions throughout the Americas, much lower than during the peak epidemic in Brazil, of course, simply because largely the high-risk communities have pre-existing immunity. So there are still minor events of and minor outbreaks, but pre-existing immunity limits the larger spread, so there's not as much transmission ongoing. But despite this, this is still an ongoing issue, simply because there's still no approved vaccine. There's actually only stuff in phase 2 trials currently, and there's also still no specific antivirals. And we were largely wondering about the regions that were thus far basically unaffected. So what about Europe? There are two concerns here. So first of all, the vector species do migrate. So Aedes mosquitoes were not really in Europe 10 years ago, and nowadays we see more and more of at least Aedes albopictus, and to a minor extent also Aedes aegypti. So there is a concern that with increasing basically climate warming also in Europe, the mosquito species will migrate and there will be new epidemics also in countries that were previously not considered to be risk countries, simply because the mosquito isn't there. And this is also exemplified by the first report that was almost a year ago, or a little less than a year ago, in France, where there was the first case of actually mosquito-acquired infections of Zika virus, where Aedes albopictus was involved. The other thing we are basically more interested in is the risk of horizontal transmission. So transmission from human to human, not necessarily involving the mosquitoes. And we were largely interested in this because simply Zika virus actually shed into a lot of body fluids. And this can, of course, open the route for a wide variety of horizontal transmissions. And I already mentioned briefly the option of sexual transmission of Zika virus. And this is actually what caught our interest or piqued our interest first, simply because there's actually an immense load of viral RNA copies in semen of infected men. So this goes up to 10 to the power of 8 copies per milliliter, which is way higher than in any other body fluid of infected people. And this is also detectable for months after infection. And this is not just dead RNA. So there was actually also in cases the infectious virus isolated, meaning there is infectious virus and very high copy RNA in semen of infected men, but still only 36 cases where sexual transmission is suspected and of course hard to verify, but at least suspected. It's also in breast milk. There are 3 cases where this is suspected to have been causing vertical transmission. And also here infectious virus is isolated. And it's also detectable in saliva, actually also more than a month after symptom onset. And the copies here also reach more than 10 to the power of 6 RNA copies per milliliter. Typically it's more than 10 to the power of 5 range, but still the RNA is there and it's also higher than in serum during viremia. And this virus has also been isolated. So the big question for us was basically, why is there no mosquito-independent Zika epidemic? Why don't returning travelers basically have a risk of infecting their partner and then eventually basically their surrounding, causing completely mosquito-independent Zika outbreaks, considering that there's such a high amount of RNA in the body fluids? And because we're basically an HIV lab, or were an HIV lab to a large extent prior to Zika, we are mostly at first we're interested in what's going on in semen, because semen is of course very important body fluid with regards to HIV transmission. So we actually already published a study on this in 2018. Just briefly want to recapitulate this because it's also of relevance for the saliva story. And to make the story very short, we discovered that while all the cells, or at least most of the cells and tissues of the anogenital tract that could be involved in Zika transmission via this route do support Zika infection, and they also support productive Zika infection, are actually inhibited by infection in the presence of semen or seminal plasma. And this is already at relatively low concentrations. So even at already 1 or 5% serum, semen or seminal plasma, which is basically semen without the spermatocytes, infection is completely abrogated for Zika. And this is not only the case for Zika, but also for the related flaviviruses, dengue and West Nile. And in contrast, very much the opposite is the case for HIV-1, which is not inhibited at all, but rather actually enhanced by the presence of semen, as you can see on the right. And the interesting thing is that actually semen contains relatively similar levels of HIV RNA copies as it contains Zika RNA copies, at least in non-antiretroviral therapy patients. So the RNA copies are similar, but in contrast to HIV, Zika is not at all transmitted via semen, or actually at a very low occurrence at least. And figuring out what's going on in semen took us quite a while. And we went through a lot of different potential molecules that could be involved in this. And eventually we found in mechanistic studies that something in semen seems to inhibit the attachment of Zika virus. Whatever the antiviral factor is, it's pretty resistant to heat and proteolysis because you can boil semen. It doesn't lose any antiviral activity against Zika. You can incubate it with proteinase K to basically digest all the proteins, and it's still completely active against Zika virus. And then eventually, after a lot of failed attempts, basically, or false routes, we eventually reached EVs, which are really, really abundant in semen. So the numbers in semen actually go to 10 to the power of 13 particles per milliliter, or even that. And we isolated these EVs, and they recapitulated the activity that we saw for full semen and also for seminal plasma, eventually leading us to the conclusion that these really, really abundant extracellular vesicles in semen are what is at least one of the major molecular species in semen contributing to inhibition of Zika virus, and thereby potentially also leading to this relatively low occurrence of sexual transmission. We also did a study on figuring out what's going on with the low transmission via breastfeeding despite RNA being present. Don't want to talk too much about this, but here we found a different mechanism that involves lipases being activated during storage and also in the of the infant, which basically physically destroy the virion. But the big question is, of course, what's going on in saliva? Again, there's 10 to the power of 6 copies infectious viruses there, but there's basically no transmission that would involve saliva, such as by kissing or something like this. So we were really curious what's going on here. So to figure this out, initially, we did experiments similar as we did with semen. So the basic workflow is above. We collected saliva from individual donors. We pooled the saliva, put this onto susceptible cells. Typically, we use VRE6 cells simply because they support Zika virus infection very well. And we basically put the saliva on the cells, add the virus immediately after, keep this on for 2 hours. and then remove, replace with fresh medium, and after 2 days determine the infection rates. And when we did this with pooled saliva, we already saw potent dose-dependent inhibition of 3 different ZIKA strains, and the inhibition is also quantitatively relatively similar for this African strain, which is called MR766, and for the top 2 strains, which are Asian lineage strains, and they are both relatively potently inhibited at 20% almost completely. And you can also see this here in immunofluorescence. You can see in green the ZIKA E protein stained cells, and blue the nuclei, and you can already see a reduction at 2.5% and the 10 and 20%. Consequently, it's relatively completely blocked. Now, a slight complicating factor with saliva is that it also has a moderate toxic effect on cells, and you can see here that at 20% on saliva, we at least have about 20% reduction in cellular metabolic activity. And that's also the reason why we do the medium change after 2 hours, simply because saliva has some factors that cells don't like too much, but you can certainly appreciate that the antiviral activity we observe is much greater than this moderate metabolic impairment. Now, already going a slight bit into the mechanism, and I will get to this in a second more, we did cell and virus treatment to figure out basically at least approximately which part of the infection the antiviral activity is directed against. So here we put either up to 10% of saliva directly on cells and then infect, or first pre-incubate the virus in a substantially higher amount of saliva, in this case up to 90% saliva, incubate this for 30 minutes and then dilute this mixture onto cells, leading to the same on-cell concentration, but of course actually higher concentration acting on the virus beforehand. And we don't see much of a difference here, so actually the curves look relatively similar, leading us at least to the preliminary conclusion here that the concentration on virus is not what determines the antiviral activity, meaning the antiviral action is rather acting against the cell and not, for example, as in the case of milk, physically destroying the virion, because then you would exceed the potency when you expose the virion to higher concentrations. Now, on the bottom, you see VRO6, and as mentioned, these are the typical cells we use for most standard infection assays. But of course, they are monkey cells, so it's a little artificial with regards to human transmission events. So we also looked at this in more relevant cells, including primary human fibroblasts and cells and HeLa cells. And you can see that this effect varies a little bit in these microscopy images, but generally, qualitatively, the antiviral action is independent of cell type. So at 10 to 20% saliva on cells, infection is inhibited in pretty much all of these cells. So we were then wondering if maybe not all donors that we collected as part of this pool. So this pool consists typically of 20 saliva donors, just so we have enough volume to do a lot of assays with it. That maybe not all of these donors exhibit this activity, and maybe there's some sort of a donor dependency. So for this reason, we first now also collected saliva from individual donors by passive druel, which basically yields relatively complex from multiple glands. Basically, the participants pool the saliva at the bottom of their mouth and then dispense it into a tube. And we analyzed this from 11 separate donors for anti-ZIKA virus activity, and also basically controlled for impairment of metabolic activity by, again, measuring ATP levels in the cells. And you can see here for these 11 donors that actually the potency by which ZIKA virus is inhibited varies pretty much quite a bit. So the IC50s go from 0.5 to more than 10% saliva, don't actually even inhibit to 50% at 10% saliva on cells, while others require less than 1%. There's a pretty drastic difference, but generally there's an inhibition in all of those samples. Now, as mentioned, this is slightly confounded by impairment of metabolic activity for some of these patients, simply because there's also differences in the salivary constituents that also affect the cellular metabolic activity. But overall, the antiviral activity is, again, greater than the impairment of metabolic activity. And there are certainly also samples which don't impair the metabolic activity at all, but still exert a potent antiviral effect. And we looked at 2 of these in more detail, namely donor 6 and 8, which potently inhibit and did not impair the cellular metabolic activity. And we looked at them basically throughout the course of a day to figure out whether the differences we see between donors are really differences between the donors themselves, or rather donation-dependent differences, because of course, saliva changes throughout the day. It correlates with food intake, with hydration status, with exercise. That is a very variable body fluid. And for this reason, we had donors 6 and 8 from this panel basically donate saliva at different time points throughout the day from 8 a.m. to 8 p.m. And at each time point, we analyzed them for the antiviral activity. And you can see that actually the variances here are relatively minor. So for donor 8, it's basically completely consistent throughout the day. IC50s vary only very little bit. For donor 6, there's some variability, but still overall, the the donor variability is much less than the interdonor variability, suggesting that whatever differences we may see might be rather due to inherent differences in the donors rather than differences between the donations. Now, also for reasons that were related to how we eventually reached the EVs in the Siemens study, and also to figure out how we can process or store the saliva so we don't have to basically collect fresh saliva for every study. We investigated how stable this anti-Zika virus activity is when we subject the saliva to different incubations and also freeze-thaw cycles. And here we basically found we can easily keep the saliva 4 degrees, room temperature 37 degrees, or even 90 degrees for an hour without really losing activity. And somewhat strikingly, perhaps, you can even boil the saliva for 20 minutes and you don't really lose any activity, as is here in the middle. And largely because it's more largely nice for usability, you can also freeze it at -80 or -20 and you don't see a loss in activity. But perhaps the boiling is pretty striking, and I will get to that again in a second. Going further with the mechanistic studies, we also found that Zika virus is reduced in attaching to the cells in presence of saliva, and which we're basically determining this by putting a high concentration of virus onto cells at 4 degrees so the virus can adhere. Then we fix these cells and directly stain the viral E protein as part of the virions, so not as newly expressed in cells, but literally staining the viral particles. And I hope you can see this through the video, but in white you can see the DAPI stained nuclei, and in green you can see virion particles attached to the cells. There's quite a lot attached at 0% saliva, and then at 20 and 60 you see virtually none attached, indicating that, as you can also see here in this quantification, higher doses of saliva reduce the attachment of Zika virus to the cells. We're using 60% here because we also have to use a very high inoculum to nicely image them, basically. So we're using a lot of virus here simply so you can see it well. And this already leads to the second mechanistic hint, because actually when you increase the inoculum, you eventually lose this effect of saliva. So just to explain, maybe for the non-virologists, so we're using here different MOIs, meaning multiplicities of infection. And then multiplicity of infection of 1 would indicate that you have 1 viral particle per cell, ideally yielding infection of all cells, at least in theory. So when you have a very high MOI, you have a large excess of virions compared to cells. And you can see here that when we infect with typical MOIs that we use for Zika virus, which are below 1, you have pretty nice inhibition here, up to 10%, but you eventually lose this when you go to 9 or even 36 MOI. And to just put this into context, so which MOIs are physiologically relevant. So the mid MOI we use here of about 2 corresponds to about 8 times 10 to the power 5 viral RNA copies in our viral stock. And this is really towards the end of what is detected in saliva. So the inhibition occurs at basically physiologically relevant RNA concentrations that would also be present in the body fluid, indicating at least that this is not, that an MOI of 36 would not be realistic because these are crazy high RNA copies. So this is pretty much the result from this was already done. Okay, so Zika saliva contains a heat-stable anti-Zika virus infector, which seems to compete for viral attachment. And this is pretty the behavior we observed for seminal plasma initially in our 2018 study, where we eventually identified these extracellular vesicles competing for attachment. So at this point in this study, we were thinking, okay, so EVs are probably the responsible factor in saliva here. And to figure out what's going on here, we first did EV enrichment. And I will get to this in a second why we did this, which is definitely not a purification. We will get to the purification also in a second. But first, we're doing enrichment, meaning we're using a protocol we call filtration, ultrafiltration, which is relatively standard filtration through 0.2 microns to get basically rid of the larger aggregates and protein aggregates and particulates that are present in saliva that are larger than 200 nanometers. And then we concentrate these by ultrafiltration through 100 kilodalton molecular cutoff ultrafiltration columns. And this allows you to concentrate and reach higher particle numbers, not guaranteeing, of course, that these are exosomes, but definitely reaching higher particle numbers. And I'll get to this in a second. And we do nanoparticle tracking analysis to figure out the number of particles and their rough size distribution. And this is how this looks like for these EV-enriched saliva, basically prepared from pooled saliva. Actually, the size distributions are relatively similar. So we have around 180 nanometers peak size or mean size for the particles, and this is slightly larger due to larger contaminants in the unprocessed saliva. And importantly, in this saliva pool, which didn't completely inhibit, at the highest dose here, when we look at particle concentrations, we basically extend the curve when we enrich for salivary EVs or for particles and reach full inhibition at this with this concentrated solution. And this concentrated solution of enriched EVs also still inhibits attachment. And more importantly, also the inverse is true. So if you basically do this ultrafiltration and then not only take the retentate, which contains your EV concentrate, but also take the bottom fraction, which is largely, if you put unprocessed saliva on top, an EV depleted version of saliva, of course also depleted of other larger proteins, but still, if you do nanoparticle tracking on this, you can see here on the bottom right, you lose almost 2 logs in particles, and correspondingly, you lose most of the activity, as you can see on the bottom right, in the smaller than 100 kilodalton fraction, which inhibits a little bit, but not quite as much as unprocessed saliva or the bottom fraction or the top fraction, which was reconstituted in the same volume. And the reason why we did this initially is because we can do this in pretty larger scale. So we again collected saliva from 11 separate donors. So these are actually the same donors I showed you earlier. But in this case, we also did nanoparticle tracking analysis on all of these and are basically plotting the inhibitory activity we see versus the particle concentration that we find in their saliva. And you can see, as with the previous data set, some inhibit completely, some of them don't. And also interestingly, the mean particle concentration we find across these 11 donors is 3 times 10 to the power of 10 per milliliter. And we did for all of these this EV enrichment by filtration, ultrafiltration, and this allows us to basically just get higher EV numbers or particle numbers containing EVs for each of this donor. And you can see, for example, very nicely for the donor 7, which is in the middle to the right, that in the salivary preparation without concentration, we reached maybe like 60% inhibition. And when we enrich EVs by this particle enrichment by ultrafiltration, we basically extend the curve again and we reach higher particle numbers, and then we completely also inhibit the infection. And this just allows us basically to go to higher concentrations of particles without necessarily increasing the saliva concentration on cells to 50 or something like this. And if you just look at the IC50 by particles here for these enriched EV preparations, we have a mean IC50 of all of these 11 donors, which is around 3 times 10 to the power of 9. So about a log lower than the mean particle concentration we measure in unprocessed saliva. Again, of course, being well aware that these are not necessarily all EVs or exosomes, but at least it gives you an estimate for the particle concentration, of which EVs are certainly a notable proportion of. Now, eventually, of course, we also want to know if purified EVs that we really purify from all the other contaminants that could be present in enriched EVs are really responsible for the inhibitory activity. And for this reason, we're applying a protocol that was developed at the Karolinska Institute in Stockholm. You might know Ruediger Gross, who's our amazing collaborator and the expert on EV purification, who helped us a lot also in the past projects and now also with saliva. And he and Manuela Gustafsson from the lab of Samuel Andalusi at Karolinska helped us greatly with purifying EVs from saliva using a combined protocol, which contains first a step of tangential flow filtration, where basically the entire sample is flown over a ultrafiltrating with a very high molecular weight cutoff. And this, first of all, gets rid of a lot of the smaller contaminants or the smaller non-vesicular particles, and also highly concentrates the sample. And then this is finally purified by size exclusion chromatography, and more specifically bind elute size exclusion chromatography with basically functionalized pores in the particle membranes, and finally concentrated by ultrafiltration. And this yields a relatively pure protein peak and RNA peak, and you can isolate EVs basically as the first protein peak that comes out of the chromatography. And we did this and also did nanoparticle tracking analysis on the purified salivary EVs in comparison to saliva. And you can see actually the size distribution is not that different from the particles. So we again have around 180 nanometer peak size. And this concentrate contains around 10 to the power of 11 particles per milliliter compared to a log lower in the unprocessed saliva. And again, the peak sizes or the mean sizes are shifted to the right for the unprocessed saliva due to the presence of larger particles that are non-EV nature. They also look like EVs in electron microscopy, and also the sizes roughly match at least what we see in nanoparticle tracking. And more importantly, this is also a real purification. As you can see here, we did a Western blot for markers of EVs or exosome-associated markers, and for salivary non-exosomal, non-EV markers. CD9 you can see as basically one of the main markers for EVs is highly enriched in salivary EVs compared to saliva in these protein normalized samples. Lysozyme, which is basically the contrary, very abundant protein in saliva, is massively depleted in comparison. Flotilin and Alix are also enriched, which are of course also EV associated. Alpha amylase is a bit more complicated because we initially also thought, oh yeah, this will also be a great control because it's non-salivary, non-EV associated salivary abundant protein. The opposite appeared to be the case. There are some descriptions of alpha amylase being associated with salivary EVs, but nothing conclusive. We're not 100% sure if this is the case or if we co-purify it simply because it's associated with the EVs. But regardless, we see by lysozyme at least that we get rid at least of a bulk of the non-EV proteins, and we certainly enrich the EV proteins. We also went a step further by looking at the surface markers on these exosomes or on these extracellular vesicles with MaxFlex, which is basically a bead-assisted technology to analyze surface marker expression on EVs. And I just want to highlight, we again nicely see CD9, 63, and 81. A lot of other markers that we also see on other EVs. One that we, at least personally, don't see a lot on other body fluid-derived EVs, I just want to briefly mention because it's interesting, is CD14, which is a LPS binding protein, which is present in saliva secreted by monocytes and has actually been implicated to be involved in basically oral immunity with regards to bacteria. For example, there's a study, a really cool study correlating that if you have low amounts of soluble CD14 in your saliva, you're more likely to get dental caries, for example. And this is actually something we haven't seen in most other body fluid-derived EVs to this content, the CD14. Of course, the most important question is, do these highly purified EVs still inhibit Zika virus infection? And they do. And they also do this after boiling and proteinase K treatment, and even benzonase treatment, which is a promiscuous endonuclease. So this degrades all forms of DNA and RNA, leading us to the conclusion that the EVs do inhibit Zika virus infection in saliva, and likely it's not due to surface-associated proteins, because they are definitely destroyed by boiling and proteinase K treatment. Not so much by boiling, but you can see on this page that after proteinase K treatment and boiling, definitely there's basically nothing left of these proteins. And it's also not due to surface-associated nucleic acids, RNA or DNA, because benzonase definitely degrades all of them and the inhibitory activity is unchanged. Now you might be wondering, and reviewers in the past have been wondering this, and we have also been wondering this, whether boiling doesn't completely destroy the EVs. And we had a look at this with TEM. So at the top, you can see untreated EVs. At the bottom, you can see what it looks like after we've boiled them. And there are definitely EVs there after boiling. We, of course, cannot be sure if these EVs are partially destroyed or at least transiently destroyed, and then they reform after cooling down again. But definitely there are particles left even after boiling. Of course, they're definitely biologically changed because the proteins might be denatured, and morphologically also slightly, but there are still particles left after boiling. Now, thus far, we've used a lot of different cell lines, but none that were relevant to potential target cells during oral transmission. So in this step, we then also analyzed primary cells that are isolated from the oral cavity. And we used primary gingival fibroblasts, which are basically fibroblasts directly isolated from the gingiva of patients. And you can see here moderately nicely a slight cytopathic effect. On the bottom, these are just normal fibroblasts. And if you infect them with an MOI of 5 with Zika, they have this relatively characteristic cytopathic effect of Zika virus, basically just cell rounding due to virus-induced cell death. But more importantly, they're also productively infected. So when you inoculate them with a low dose of virus and then watch what happens in the supernatant over a few days, you definitely see infectious virus being shed. So they are productively infected, meaning there are definitely cells in the oral that could be infected were it not for antiviral factors blocking it, for example. And also in these cells, the salivary EVs exert their inhibitory activity on them, basically leading to a complete block also at concentration similar to what we saw on the other cell lines. Now also, especially with regards to what we already published with the semen EVs and what I told you earlier about the semen EVs, you might be wondering if all EVs or maybe even all lipid vesicles exert this effect, so if this is completely unspecific. And to ensure you that this is not completely unspecific, and also to ensure ourselves that it's not completely unspecific, we also used synthetic liposomes, so just phosphatidylcholine neutral liposomes. And these you can put onto cells at concentrations exceeding 10 to the power of 12 per milliliter, and nothing happens, showing that at least it's not a pure lipid deposition effect, so not simply vesicles being in the way of the virus. But then we also isolated EVs from different body fluids. And of course, we're largely interested in body fluids that could be involved in a potential sexual transmission scenario. So we isolated EV from semen, saliva, and vaginal lavage with this combined TFF-BE-SEC approach, and also enriched EVs from urine with filtration, ultrafiltration. And you can see that they do inhibit Zika virus, but the inhibitory activities are pretty drastically different. There's more than a log-in difference between the salivary EVs and the semen EVs. And of course, you also have to consider at this point that the concentrations of these EVs in the respective body fluids also differ drastically. So in semen, as mentioned, we're exceeding 10 to the power of 13, while as I showed you earlier in saliva, we're almost 3 logs lower. Considering this, of course, the salivary EVs are even way more potent in physiologically relevant concentrations that are actually present in the body fluid. And then secondly, you might be wondering, so is this a completely unspecific effect with regards to viruses? Do all viruses get inhibited by this? And for this reason, we looked at SARS-CoV-2, which, like Zika, reaches pretty high titers in saliva of infected individuals. And of course, this is also exploited, as you all know, for diagnostic purposes. So people are also thinking of doing salivary-based diagnostics instead of the nasopharyngeal swabs. And actually, the RNA copies are not that dissimilar. So the median RNA copies you see for SARS-CoV-2 patients in saliva are also at the range of 10 to the power 5 copies per mil. But unlike Zika virus, SARS is, of course, highly contagious and also transmitted in the presence of saliva containing respiratory droplets. So for this reason, we were pretty curious to see what happens when we do basically the exact same experiments in presence of saliva or salivary EVs with SARS-CoV-2. And the difference could not be more stark. So we have here again 10 separate saliva donors and a pool of it on the left, and reaching concentrations of up to 10% saliva on cells. And you can see on the top, I'm sorry, there's a label missing. Top is Zika and the bottom is SARS. So on the top, you can see that all of the saliva donors potently inhibit Zika virus infection, reaching almost complete inhibition for most of them at 10%. And on the bottom, which is basically the same experiment with a French strain of SARS-CoV-2, we don't see any inhibition, even at 10%. If anything, we see a slight enhancement, but definitely no inhibition of infection. And also the purified EVs, they don't do anything, as you see on the bottom right. So even at 2.4 times 10 to the power of 10 particles per mil, which is well enough to inhibit Zika virus infection, we don't see any inhibition of SARS-CoV-2 infection inhibition. It leads us to the hypothesis / conclusions that extracellular vesicles, which are abundant in a lot of body fluids, represent at least one mechanism of innate antiviral immunity that contributes to restricting the horizontal transmission of certain viruses, which are at least present in these body fluids, but not transmitted. And what we see, at least in our studies, is that this basically matches the modes of transmissions these viruses use. So HIV is present in semen and also transmitted in semen, but not inhibited by semen EVs, unlike Zika. And basically the same is true for SARS and saliva and Zika. And of course, we're not the only ones describing antiviral activity of body fluid-derived EVs, but in the case of Zika, this I think constitutes an interesting mechanism. And I showed you today that saliva contains a lot of these EVs. The mechanism involves competing for cellular attachment, and that the potency is donor-dependent and probably really donor-dependent and not donation-dependent. And that EVs from other body fluids also do inhibit, but there are drastic differences in potency with regards to how many particles you need to inhibit infection by 50%. But we think that at least in saliva and semen, these EVs in both body fluids at least offer a partial explanation to why there's not more of a mosquito-independent spread of the virus. And what we're currently still looking at is how viruses are affected not only by salivary EVs, but also by EVs from complete body fluids. I mean, basically every body fluid contains some EVs and also some viruses. And I think it will be pretty interesting to eventually unravel how viruses are transmitted in body fluids containing EVs and what the interactions here are. We also want to figure out what the donor dependency is based on and how the different potencies of EVs basically are, what they come back to. So what the constitutional differences in the EVs from the different body fluids are and how this explains the potencies. And of course, the most crucial question is what is the exact mechanism of inhibition and can we exploit this for translational purposes? And I have to disappoint you that I can't tell you this completely today yet because we're still working on it. We have some hypotheses on this and we're still in the process of filling it. And I cannot speculate too much on it, but we're definitely working actively on this and you can look out for updates on this by us because hopefully we'll have something more conclusive soon. but we're definitely very interested, of course, in the mechanism and how we can exploit this therapeutically, maybe also for other emerging viruses. So with this, I've already reached the end, and I want to acknowledge and greatly appreciate the people doing the work also in Ulm together with me, most importantly, our director and my basically my mentor, Jan Münch, and of course my great colleagues, Karina and Janis and Franziska, and most notably also the people from Stockholm who are the experts who basically introduced us to how to really purify EVs, Andre Gergens and Manuela Gustafsson in the lab of Samuel Andalusi, and giving us the money to do all this work, which is the European Commission, the DFG, and other different institutions. And then I, of course, thank you all for the attention, and I'm really happy to discuss and take questions.
Well, thanks very much, Ruediger, for that presentation. I think this is fascinating work, and we've certainly had a lot of questions that have come in the chat box already. Yeah, and that's great. It looks like you have a lot of young lab members there, so it's good.
This might be biased by the fact that it was from a Christmas party.
Anyway, so I'd like to first go. So we'll go through the list of questions that we have here. So starting out with some questions about the basic biology, I think, of Zika virus. So I'm going to unmute Stephen Amoa. Could you ask your question here? And am I able to unmute? Maybe not. Oh, there we go.
Hi,
Hi.
Great talk. I mean, I have a background in exosomes, so I was really fascinated that I have this opportunity. So I was wondering, but I think you've really answered some of these questions unless you want to shed a bit more light. So while speaking about the presence of Zika virus in semen, I mean, I was wondering whether you'd also find more within the spermatozoa or just in the fluids, and also about their presence in vaginal fluids. But I think you answered some of this. So if you could maybe elaborate a little more on that. Thank you.
So you're wondering if actually the cells in sperm, in seminal fluids, are actually the spermatocytes, whether they are infected, right?
Yes,
so I don't
correct.
think this case. I think I might be speculating, but I don't think they can be infected by any virus because there's really not any replicated machinery going on in these. So at least certainly not with Zika virus. So to my knowledge, there's no infection going on of spermatocytes. And the second question was with regards to vaginal fluid. I think there are relatively few descriptions of Zika in vaginal secretions, but this is of course also much harder to analyze than semen. And I think there's not really any, I don't think from vaginal secretions there was isolation of infectious virus thus far. There was also presence of RNA that I know, but not a lot of studies being done on this. And I don't think there was infectious virus isolated from vaginal secretions.
Great, thank you. Next question is from Antonis on lipase activation.
Mm-hmm. So this is regarding the breast milk study.
Yeah, hello, Ruediger.
Hi.
Can you hear me?
Yeah.
Yes. Nice talk. Thank you very much for this and for the information that you provided. I just have like a theoretical question because you mentioned that there was a graph about the lipase activation and the production of yeah, it was at the beginning.
Yeah. Oops, no, I was too fast.
Yeah.
I get to it.
Yeah,
you can ask another graph.
The next one, or I can maybe
yeah, finally. Yes.
That produces you said produces bonded antiviral fatty acids destroying virions, and yes. I was wondering if there is like a known mechanism or you know something about this because it's quite interesting, but I don't understand what exactly can happen there.
Yeah, so that's a pretty cool subject. Actually, we didn't that far into the details because there's also other studies by Stephanie Pfender and other people in Germany. I can also send you these studies. So this was also already done for HCV, so hepatitis C virus. And the hypothesis here is actually that fresh milk, it doesn't do anything. So if you have fresh milk and put your virus in, at least for Zika and also to some extent for HCV, nothing happens. However, when you put this milk at 4 degrees or and potentially also in the infant stomach, but I have to extend a bit for this. Something happens where basically lipases move from the protein fraction to the lipid fraction in milk. And this happens both at cooling, basically during cooling of milk, and also in the stomach of the infant, whereby these lipases move to basically where the lipids are, and they cleave free fatty acids from the larger lipoproteins on the lipid globules. And these form micellar structures, and they literally act like a detergent on the very end and physically by the very end. And in the case of HCV and of ZIKA, you can see this by just looking for the release of viral RNA, which definitely happens. So the viruses are physically destroyed. And this happens maybe via similar mechanisms when you cool the milk, because there are two lipases in breast milk. One migrates to basically the lipid phase during cooling by mechanisms not fully understood. And there's another lipase which becomes activated by bile acids in the stomach, and then also starts cleaving these free fatty acids. But basically, there are two lipases in breast milk. They activate in the stomach and also during cooling, they cleave free fatty acids, and these form micellar structures, and they physically destroy envelope viruses. And this is described for ZIKA and for HCV.
Okay, thank you very much. Thank you.
Sure, thank you.
Thanks. So we have a question here from Herman Outmeppen, who asks about the potential molecules that are actually involved in this. Since you've treated with proteinase K, like what could it, I know you said that you don't have those data yet. You don't know for sure, but can you speculate on what they might be?
Yeah. So this obviously is a complicated question, because a lot of molecules are excluded by the fact, of course, that proteinase K treatment and boiling doesn't do anything, and that it's an attachment effect. So even you could, of course, argue that, okay, there's cargo that is protected during the boiling and also by the proteinase K treatment. But that kind of argues the competitive aspect. So clearly something is competing for attachment, which could either be relatively unspecific attachment that is used by ZIKA, or maybe even integrins or proteoglycans or something on the cell surface, or it could be more specific molecules, lipids or specific peptides, which are maybe not as affected by the boiling or the proteinase K treatment. But also because of the differences between the body fluids, which we're still investigating, I can't really give you a certain answer on this, and I will have to unfortunately delay this answer for a few weeks or months until I can really tell you for certain.
Okay, so Herman, any follow-up on that?
No, I thank you very much. Greetings from the German north to the south, and very nice presentation. Thank you.
Thank you.
Great. Okay. So I really like this next question, which is from Krishna, who has a couple of questions that I find really intriguing. So I'm unmuting you now if you would like to go ahead and ask this.
Hi. Thanks for the nice talk. So my first question is related to have you observed from the different ethnic populations, like more susceptible populations, or your patients like cohort also contains from the disease individuals? So whether their saliva is different. And my second question is like, what is the difference in the species, like from the mosquito saliva, or like the common animals like monkeys, which act as the virus loads too. So whether that those saliva also produce the same kind of effect you observe? Thank you.
Thanks. I agree, Ken. These are great questions. And actually, we were also, so of course, I mean, we're somewhat biased here because we're in Europe and there's no dengue and no ZIKA really in South Germany. And all our samples come from people in the lab, basically. There's kind of a spread of the age, but all of these people are basically naive with regards to most flaviviruses. We actually include a table of the travel history and vaccination status of these donors in the paper. So none of them have had any serious flavivirus disease. Some of them are vaccinated against yellow fever, and most of them are vaccinated against TBEF, not all of them. There's not really any correlation that you could make based on the fact that these people had TBEF and they neutralize more efficiently. So there's really no conclusive info, at least from these around 20 or so people that we analyzed. And we haven't looked at diseased saliva. Kind of hard currently because there's not as many ZIKA cases, especially here. But I agree, it would be interesting to see what actually happens when you actually have also the virus in saliva. But unfortunately, we haven't done that. With regards to monkey saliva, or at least animal saliva, I think you also asked about mosquito saliva. This is we actually also were thinking of looking at this, and we have collaborators that were telling us, okay, they add us with a microliter of it. So mosquito saliva is a very rare commodity, so we haven't really done this yet. But I agree, this is actually really something I'm interested in. So if you have a resource of large volumes of mosquito saliva, we'd be happy to look at it, but unfortunately it's really complicated. But I agree, this will also eventually, I think, be our goal to also look at how this is different in different species, especially with monkeys where saliva is more easily attainable. But unfortunately, can't really give you a great answer yet.
Yeah, I would hate to have that job of having to express the saliva of a mosquito.
We were told a microliter is possible. I don't know how they take it out.
Yeah. Interesting. Good. Okay. Let's see here. We have a question from Shiv about the characterization. Shiv, do you want to ask this? I've got to see if I can unmute. Sorry, I'm technically not the host today, so I'm not unmuting everybody. There we go.
Yeah. Hi, Ruediger. Nice presentation. I have just one question. Did you also characterize SARS-CoV-2 EVs by anti-A electron microscopy or by EV markers for Western blotting and MaxPlex that you showed for Zika virus EVs?
What do you mean? So EVs secreted by cells infected with SARS?
Yeah, SARS-CoV-2.
We can, or have we have done it?
Okay.
I don't know. So what was the question? If we can do it or if we have done it? So we haven't done it. We could do it in theory. The question is which target cells would be the most relevant to do this from. And yeah, I think it would be interesting to see basically how the surface markers change and stuff like this. But we haven't done it, no.
Thank you.
Yep, great opportunity for more studies. I've followed up with Mona. Mona Batish has a question, but we've agreed that this is, this is something that we're going to have to wait for your next paper for. And so I'll move to Shannon Pendergrass, who asks, let's see, Shannon is here. Shannon, unmuting you now. Go ahead.
Hi, great talk. I know that the saliva has a lot of bacterial OMVs that co-purify with the human EVs. And these OMVs are coded in LPS. I'm wondering if maybe LPS is the inhibiting factor, as LPS has been shown to inhibit other viral infections.
This is a great question. What would the molecular weight of the LPS be? It's also pretty big, right? But would you think it would co-purify really with the EVs after size exclusion and DFS?
Well, bacteria produce extracellular vesicles. They're called OMVs, and those are about the same size as mammalian EVs, and often co-purify in saliva and urine preps. I also know, saliva is one of the most potent biofluids. One of the difference between EV preps from saliva and other biofluids is just the amount of contamination of OMVs.
That's actually an excellent question that I've largely also so far considered with the vaginal lavage EVs, because I would also expect, at least there, I would expect tons of lactobacillus-derived EVs to be present. But so I think this actually, so we haven't looked at this in detail, but you're right, that this could at least play a role and also impact the potency differences, because in semen, at least, we wouldn't expect any bacteria-derived EVs. And of course, semen EVs do something. And I agree, it would be interesting to look at maybe if we can get these bacteria-derived EVs in absence of the human-derived EVs in the saliva and see how much they do. So we haven't looked at this, but it's a very good suggestion.
So also, it's very tricky to separate OMVs from mammalian EVs. Maybe the first thing you could do, which would be much easier, would just be to find some enzyme that destroys the polysaccharides and try that.
Yeah, that's a great suggestion. Thank you.
Yeah, that is a really good suggestion there. So thanks a lot, Shannon. We have a couple of comments here too. So Lucia Languino wants to mention that there was a study that came out recently on EVs from vaginal fluid and how they could help to block transmission of HIV. And indeed, my lab also has a recent publication where we looked at a particular microRNA that seems to be acting in an anti-HIV manner. So there are numerous candidates I think that you would have to follow up on, depending on what your profiling results turn out to be. And then Alara Cohn also states that Zika virus has been shown to replicate in testicular tissue. So this comes back to the early question about the biogenesis. I would like to ask a few questions myself, because I think there's been a lot of discussion in the community recently, particularly amongst labs that might not have access to BSL, level 3 facilities or whatever the equivalent is around the world, to study SARS-CoV-2, for example, directly, or for that matter, any virus that has to be handled at a biosafety level. What are some ways that we can? You mentioned heating, but what are some ways that we can inactivate viral components in biofluids without affecting EVs or without, I guess, destroying EVs? Are there any other things that you've done?
Yeah, that's an excellent question. So, I mean, we don't really, so for SARS-CoV-2 is obviously a hurdle, a bigger hurdle. We also didn't have a full BSL-3 lab prior to this pandemic. So the highest BSL pathogen we worked with was also HIV. And I don't know how it is in the States, but here it's basically a reduced BSL where you can do HIV stuff in. So this is like a BSL-3, but without the air filters. And we also had to basically join another lab that has a BSL-3 to work on this. And we really do basically as much as we can in there and only take stuff out that is heavily fixed and completely inactivated and verified to be inactivated. But typically, the most gentle method, and it depends on, of course, on your downstream processing, that we would have used normally also in the past for virus inactivation is just UV, because this at least doesn't alter your stuff too much. Of course, it functionally kills EVs also. But I mean, UV heat, but this also kills EVs to some extent, at least if you're looking at stuff that is more delicate than what our mechanism might involve. I mean, you could also try to basically block your virus with antibodies, but I don't think anybody would be fine with taking antibody-blocked virus out of a BSL-3. So I don't know if there are any other virologists in the crowd that could weigh in on this, because I definitely don't have the definite opinion on this. I don't know if you have a better opinion on it, Ken. So,
well, I mean, I think there are a lot of potential ways to do it. It's just that it doesn't seem that there's a strong literature on how EVs are affected by these things. I mean, it's actually relatively a new thing in the world that we're worrying about EVs versus viruses, and finding that, in many cases it's quite difficult to separate them. It can be done for a lot of viruses with density gradients, but of course that would also have to be done at that at that BSL-3 level for certain viruses. And if we could inactivate it and still recover some kind of property of the EVs, even if it's not function, but just the ability to characterize them, that would be incredibly useful. So, I mean, I've looked through the literature. Some other people in my group have looked through the literature, and I have collaborators who have done it too, looking at recent studies on SARS-CoV-2. And it does seem that to reduce viral titers down to almost undetectable levels, there are fairly harsh treatments that are required.
Yeah, exactly. I mean, the antibody stuff was just something I was thinking of. I mean, if you really, really have a super potent neutralizing antibody and you put it on a high concentration, probably you could reduce the infectivity completely, but I don't think any regulatory agency would be fine with saying, okay, this is inactivated now, you can take it out. So functionally, probably, but I wouldn't be fine with taking it out. And that's the reason why we do the harsh treatments, to be really, really sure. That's the problem.
Right. Okay. All right. Were there any other comments? I didn't see anything else coming up in the chat box about that particular question, but if anybody has some insights, I'd love for you to weigh in on it. And if not now, please feel free to get in contact with me and Ruediger later, to let us know if you've had experience with inactivations that would not necessarily destroy everything about the EVs. All right. So I think that brings us to the end of our questions for today. So I'd like to thank everybody for joining, and for the nice discussion that we've had too. I think has covered a lot of different topics today. And most of all, thank you so much, Ruediger, for joining us. We really appreciate your sharing your paper, and we wish you all the best as you continue with learning more about the mechanisms here. So thank you, everybody, and we look forward to seeing you again soon.